Okay, so welcome to the second session of the Katz Symposium.
She stopped.
Our first speaker is going to be Jade On.
Hello.
And Jade, yeah. Jade gets connected.
Hello. Hello.
The title of Jade's talk is The Role of Microtubule Cytoskeleton in Limiting Sensory Hair Cell Regeneration. Jade?
Hi, so my name is Jade, and I'm going to present the comprehensive research that I've done in the Corwin lab in investigating the sensory hair cell regeneration. So as everyone knows, the inner ear is an incredibly important part of the human body. It's not only involved in hearing, but also maintaining balance and equilibrium throughout the entire body and detecting the movements and accelerations of the head. And anatomically, the inner ear is composed of two major components. You have the spiral-shaped cochlea, which is primarily involved in hearing. And you also have the vestibular system, which is this region right here, and it has three major organs. You have the utricle and the saccule, which would be localized in this region, and the semicircular canals, which is right here, and that's involved in balance. And it's been shown that hearing loss dramatically increases as you age, which is expected because sensory hair cells don't regenerate after they die. And not coincidentally, there's also a decrease in both inner and outer hair cells as you age, and this is associated with hearing loss. So if you're wondering why I chose to study sensory hair cell regeneration in the inner ear, you have 30 million Americans that have hearing defects due to the loss of sensory hair cells. And this is primarily due to the fact that mammals cannot effectively replace their damaged hair cells just embryonically, although regeneration does occur quite readily in non-mammalian species like birds and amphibians. And investigating the processes that are involved in restoring these damaged hair cells, as well as determining possible means for preventing their loss, can not only help in alleviating disorders that are due to balance and hearing, but also potentially other neurosensory disorders like Alzheimer's and blindness. So hair cells are incredibly sensitive to damage through environmental traumas like ototoxic compounds, cerebral trauma, and just basic exposure to loud noises. However, there are a few models for sensory hair cell regeneration. And in the regenerative cellular proliferation model, following the death of a hair cell, you have an adjacent supporting cell that re-enters the cell cycle, divides or replicates its chromosomes, divides, and then produces cellular progeny that differentiate into hair cells and supporting cells. And these replacement cells typically regain functionality within 30 days. There's also a cellular phenotypic conversion model. And in this model, you essentially just have a transdifferentiation of supporting cells into hair cells. And if you're also wondering why I'm looking at the cytoskeleton, it's an incredibly important structure that's found in essentially all cells. And it's involved not only in maintaining the shape of the cell, but providing a means for locomotion, transport of organelles, and providing rigidity to the cells. So my research, my lab focuses on the sensory epithelium of the utricle. And the images you're going to see are from a publication that was written by a graduate student in my lab. And it shows that in non-mammalian species like the F-actin belts remain thin with age. So here it's when the chick is born, and this is a year after it's born, and the F-actin belts are still thin. However, this is not the case in mammalian species like mice, where you have an increase in the width of the F-actin belts with age, and this increase likely plays a role in restricting sensory hair cell regeneration. So to start out my research, I wanted to find out where the microtubules were localized. And to do this, I first had to culture the sensory epithelia, and then I performed immunohistochemistry and cryostat sectioning. And again, where do the microtubules localize? This is the sensory epithelium of a post-embryonic day 25 mouse. And here I've stained for microtubules using alpha-tubulin. And these are, right here you see supporting cells, and these well-defined circular cells are the hair cells. So alpha-tubulin in the red channel labels the microtubules. Phalloidin labels the F-actin belts, and the last panels here show a merge of the two channels. And it's clear that at both the apical surface on the top and adherence junctions on the bottom, the microtubules are localized within the supporting cells primarily. So after this, I wanted to figure out what the orientation of the microtubules was relative to the supporting cells and the hair cells, so I performed cryostat sectioning. And here you have images from two different post-embryonic day 34 utricules. Again, in green, you have labeling of F-actin. And the blue is myosin 7A, which is labeling the hair cells. And in red, you have microtubules. And this told me that the microtubules were running down the length of the supporting cells. You can see these very distinct lines running down. And they were also localized in the necks of the hair cells. However, I couldn't definitively state anything from these pictures because the resolution wasn't good enough. So I next wanted to see if there were ways for me to resolve the resolution limitation from cryostat sections. So to do this, I used freeze and dry cracking and subsequent analysis with scanning electron microscopy, and also looked at images of the sensory epithelium of the utricules using transmission electron microscopy. So these are 3 images taken of the sensory epithelium. And it essentially shows the three-dimensional substructure of the sensory epithelium. As you can see here, here's a hair cell, and here's a supporting cell. But you can't actually see the microtubules in these images because the cracks that I produced weren't intracellular. So to see if I could actually see microtubules, I also used transmission electron microscopy. And here you have a transverse view of a post-embryonic day 0 utricle sensory epithelium of the utricle. And I zoomed in to an image of a supporting cell. And I don't know if you guys can see, but there are bands of microtubules that are discontinuously arranged within the periphery of the cell. And these rings that were around the cell were primarily characteristic of only cells that were very circular in nature. And I also looked at the sensory epithelium of a post-embryonic day 53 utricle. And this is just showing a longitudinal view of a hair cell. And it's easier for you to see the microtubules here, but they're running in distinct bands down the hair cell. So both scanning and transmission electron microscopy increase the resolution of the images that was previously limited by the cryostat sections, and scanning electron microscopy allowed for a visualization of the three-dimensional substructure of the sensory epithelium, while a transmission electron microscopy allowed me to look at the arrangements of the microtubules in the cells. So after doing that, I went on to look at, I went ahead and ran experiments to test one of my hypotheses, which was that the microtubules within the supporting cells get heavily stabilized with age. And to do this, I performed fluorescence recovery after photobleaching on YFP tubulin transgenic mice and also nicotazole drug trials. And nicotazole is a drug that inhibits the polymerization of microtubules. But I'm not going to show that data today because of time constraints. So to answer how stable the microtubules were in the cells, like I said, I performed FRAP, which involves exhausting the excitability of a region of interest and watching for the recovery of fluorescence in that area as an indication of the stability of, in my case, microtubules. So this is a video of FRAP that I performed on the neck of a hair cell. Can you run it again? Right here. So I exhausted the excitability and then watched it recover. And it recovered pretty quickly over the time period that I watched it, which indicates that the hair cells within the sensory epithelium are pretty dynamic. And I don't have a video of this, but I also photo bleached a supporting cell. And in that clip, I didn't see much recovery of fluorescence, which indicates that the supporting cells are much more stabilized than the hair cells. And that might contribute to why humans can't regenerate their hair cells, because if the supporting cells are so stable, they probably won't be able to differentiate into new hair cells. So in conclusion, while the research methods that I used allowed for an effective visualization of the microtubule cytoskeleton, given the time allotted, it only allowed for a partial analysis in answering my question. And while there is a possible correlation between the microtubule cytoskeleton and the reasons why humans can't regenerate their inner ear hair cells throughout their lifetimes, that correlation is currently unknown. I don't have time to go over research topics, but there are plenty. And I just wanted to acknowledge the members of my lab, Dr. Corwin, Joe, Ben and Sol, who aren't here. I couldn't have done all this without you guys.
You mentioned the cryosectioning caused poor resolution for yours. So why is it that method of sectioning degrades resolution compared to something else?
That's the only type of sectioning that I performed, and it wasn't the method, it was more of how thick my sections were. So I cut the sections at 16 micrometers, and usually to get really high resolution, you should be around like 1 micrometer. So it wasn't the method, it was me. Yes.
I didn't see individual scales in your micrographs. What's the diameter of microtubules like?
Microtubules are on the scale of 25 nanometers, and on my transmission electron microscopy images, I performed a measurement using ImageJ, and the diameter of those tubes were around 23 to 24 nanometers. So that's what told me that those were microtubules, as opposed to, F-actin bands, which are around 8 nanometers.
What was the thickness of the EM sections?
the transmission electron. I don't remember, unfortunately.